Welcome everyone. This is a supplemental lab video for Bio 137. This video goes along with the first of our histology labs. Histology is just the study of tissues. So we're going to be looking at several different tissues today. Most of this will be the more common epithelial tissues. And then after the epithelial tissues, we will be looking at connective tissue proper, which are the connective tissues other than bone, blood, and cartilage. There's going to be 12 slides that we look at in this video, and keep in mind that this is just a supplemental. There is no substitute for actually being in the lab or the open lab and looking at these tissues through a microscope. You definitely want to have that experience. You want to get used to using a microscope. You want to see these tissues with your own eyes, because in this video, I'm showing you some examples, but something that you'll learn really quickly in this class is that every time you look at one of these tissues, while there's going to be some stuff in common, every slide will look a little bit different. So this video is really just to point out the features that you should be looking for as you look at these tissues. And as we look at these tissues, there's a couple of things that you need to keep in mind. First, the color of these tissues is not the color of these tissues while they are in your body. When making a slide, whoever's preparing the slide stains the tissue. And different stains will produce different colors. And while it's really common for certain tissues to always be stained pink, that's not a rule that's set in stone. So pay no attention to color when you're learning these slides. Just because the one that you're looking at happens to be pink, or the one in the example happens to be pink, and you look at your neighbor's slide and it also happens to be pink, don't learn, oh, the pink slide is such and such. Because when it comes exam time, maybe the slide used on the exam will not be stained the same color as the one that you look at. So instead, learn shapes, learn features, because those will be constant. Those will not change like color can. And another thing to keep in mind is when we look at tissues in the body, very, very few locations will only be made of one single tissue. So as a result, when these slides are prepared, there's almost always going to be more than one tissue on that slide. So when you're looking at one of these slides and you're looking for, say, simple cuboidal epithelium, but you don't see anything like what the picture is, it's very possible that you're looking at a different area of the slide and seeing a completely different tissue. So these pictures, either in this video or in your lab manual or in the textbook, those are pictures to give you an idea of what to look for when you're trying to find this on a slide. So let's look at our first tissue example. This slide is going to show us an example of simple squamous epithelium. Now, the slide itself, this comes from the lungs. This shows the air sacs or the alveoli inside your lungs. But we can find simple squamous epithelium in several places through the body. This just happens to be one example. Now, anytime that we hear simple when we're looking at tissue, it means that the tissue is only one single cell layer thick. And here the second word, squamous, that's a word that means flat or squashed. So simple squamous epithelium, the cells are going to be really, really flat, and the nucleus tends to be kind of elongated rather than round. So on this slide, here is an example right here. You can see the cells are really, really flat, and this kind of dark dot right there, that's a nucleus. Up here's another good example. The cells are really, really flat, and we can see the nucleus right there. There's a few other examples. If we look on here where we see really good evidence of simple squamous epithelium, but scattered here and there, you'll see cells that are not so flat and the nucleus is pretty round. That's what I was just talking about. Here and there, you will see some tissues on this slide that are not simple squamous epithelium. But most of the cells that we see on this slide are very, very flat, and the nucleus is kind of elongated. So they are simple squamous epithelium. Now, simple squamous epithelium, since it's so flat and so thin, only a single layer of really squashed cells, it's not very thick at all. It's easy for stuff to get through, and that's the function of simple squamous epithelium. It makes diffusion of stuff really, really easy, really efficient. Like we see in our lungs, we want the oxygen and the carbon dioxide to move in and out of our alveoli, those air sacs, really easily. In our kidneys or in our capillaries, we need stuff to be able to get in and out of your blood really easy, really efficiently, and that's where we find simple squamous epithelium. Our next tissue type is simple cuboidal epithelium. And again, remember, when we hear the word simple, it means it's one single cell layer thick, and cuboidal describes the shape of the cells. So in this case, cuboidal doesn't necessarily mean it's a perfect cube, but it tends to be about the same height as the width. So rather cube-like. So we're going to be looking for something about like what I'm drawing right here. And the nucleus of simple cuboidal epithelium tends to be pretty round, pretty spherical, not stretched out the way that we saw it in the simple squamous epithelium. So something kind of like what I've drawn here. Now, this slide is a perfect example of what I mentioned previously. There are several different tissue types on this slide, but we can see lots of examples of simple cuboidal epithelium. All of these ring-shaped structures. These are all simple cuboidal epithelium. But a lot of students come up with the question of, how is this a single layer thick? Well, what we end up with is, let's look at this ring right here. We can see inside there doesn't appear to be anything there, and that's the case. This is hollow. And then around the outside, there's nothing there. This part is kind of not completely hollow, but kind of hollow. So there's nothing on this side, there's nothing on this side. This is a single layer thick. It just happens to be rolled into a ring. So one layer thick, and the cells are roughly cube-shaped. Simple cuboidal epithelium. Now what we're looking at right here is a part of the kidney. These are sections of something called kidney tubules. And this is part of your kidney that is involved in making urine. And we find simple cuboidal epithelium in places where we see the activities called secretion and absorption. Either something is moving out of these cells or something is moving into these cells. Simple cuboidal epithelium is responsible for secretion and absorption. Our third type of epithelial tissue is simple columnar epithelial tissue. Simple, again, one layer thick, but columnar, the cells are very tall and skinny like a column. So in this case, what we see is something kind of like this. And usually on simple columnar epithelium, the top of it has something called microvilli. And we do see that here, I'll point out in just a moment. But what we see in this case are really tall and thin cells, and one side of it kind of appears a little fuzzy. And scattered in this tissue, we often see something called goblet cells. And goblet cells are shaped kind of like a goblet, like a wine goblet, which is what we see here. So here's our layer of simple columnar epithelium. We can see along the edge of it this kind of fuzz. That's our layer of microvilli. And here is a really good example of a goblet cell. And here's another example and another example. Goblet cells tend to not take on stain very well. They tend to be kind of clear while the surrounding tissue takes on the stain, in this case pink. So again, we see other tissue types. It does look like this is many layers thick, but only this layer right here is the simple columnar epithelium. Down here is something completely different, so it's not considered a part of that epithelial tissue. The epithelial tissue is only one cell layer thick. Simple columnar epithelial tissue also is involved in secretion and absorption. A lot of times what is being secreted is the mucus, and that's being secreted by those goblet cells. This next tissue doesn't really follow the standard naming scheme. It's actually got kind of a weird sounding name. It's called pseudostratified columnar epithelium, or sometimes it's written as pseudostratified ciliated columnar epithelium, because it does typically contain cilia, like in our example here. And the name actually has some meaning behind it. When it says pseudostratified, the root word pseudo means fake or false. And stratified, like we've seen already now, means, multiple layers. So if we look at this tissue, or this slide here, there's actually several tissues. What we're concerned with is this top area right here. This is our pseudostratified columnar epithelium. And if you look at it, it looks like it's actually several layers thick. When we look right here, you can see, there's a nucleus, and there's a nucleus, and there's a nucleus, and there's a nucleus. So it looks like there's a couple of layers there at least. But if you look really, really closely, what we're actually seeing, if we look over here at this drawing that I have on the left, it's one layer. All of the cells rest on this same area right here. This is called the basement membrane. Every cell there sits on that basement membrane. Some of them are a little wide at the bottom and narrow at the top, and its neighbor may be wider at the top and narrow at the bottom. So it kind of looks like there's two layers or more there, but if you really look closely, they all sit on this same basement membrane, so it's only one layer thick. It's falsely stratified, pseudostratified. And something that we will see here that we saw in the previous tissue are goblet cells. These big clear or mostly clear cells, those are the goblet cells. And just like in columnar epithelium, these goblet cells produce and secrete mucus. So that's one of the functions of pseudostratified columnar epithelium is mucus secretion. But if we look here along the surface and kind of what I've got drawn along the top here, we see cilia. And as that mucus is secreted, these cilia move the mucus along. Remember from the chapter 3 lecture video, cilia, when they move substances across the surface of the cells. So one place where we would find these is in the respiratory tract, especially down through your trachea. And your trachea, sometimes called your windpipe, when you breathe in and there's dust or other debris, it gets caught in that mucus, and then the cilia move the mucus upwards towards your throat so that you can cough it out. Now, if you're a smoker or know someone who's a smoker, smoking burns those cilia off and you end up just inhaling any debris. It doesn't get caught, it doesn't get propelled upward. So this is, again, pseudostratified ciliated columnar epithelium. Now here's our first actual stratified tissue that we're looking at, and we're looking at the dark purple in this slide. This is stratified squamous epithelium. Remember, stratified, more than one layer. Squamous, the cells are squashed or flat. And if we look along the surface here, we can see lots and lots and lots of layers of really flat cells. So this is that stratified squamous epithelium. Now, there is a part of this that doesn't really look completely flat, and that's deeper down here. And really, these are cuboidal cells, and they're special. They're stem cells that are actually dividing to produce all of these cells at the top. So really, the cells at the top, that is the stratified squamous epithelium. And those cells are either dead or dying off. And that's what we want. It's not a bad thing that they're dead or dying, because the purpose of this tissue is abrasion resistance. It flakes away. For example, we find this on the surface of our skin in the epidermis, which is the outermost portion of our skin. We also find it lining the mouth, lining the esophagus, places where there is a lot of abrasion. Think about what happens when you scratch your arm. As you scratch your arm, you're flaking away these outer layers. Not a big problem because, like I said, they're dead, so you're not hurting anything. And those are constantly being replaced by these stem cells deeper down. So stratified squamous epithelium, many layers of really flat cells found in places like the epidermis, the outermost layer of the skin, the lining of the mouth, the lining of the esophagus, and it's there for abrasion resistance. This is the last of the epithelial tissues we'll look at. There are other types of epithelial tissue in the body besides the ones we've looked at. We're just looking at those that are the most common. So this last type, it is a stratified epithelium, but it doesn't have that in the name. This is called transitional epithelium. And if we look at these cells here, some of them are columnar, some of them are cuboidal. If this were to be stretched, this white area here is a hollow part of the structure we're looking at. This is actually something called the ureter. This is what transports urine from your kidneys down to your urinary bladder. As urine passes through, this tube stretches and this white area right here expands. And as we stretch these cells here, they go from this cuboidal or columnar shape into more squamous shaped. And because of that, it allows things like your ureter and your urinary bladder to stretch, to expand, and then to snap back as they're emptied. And while it is not good to learn, I guess cheat isn't the best word to use here, but kind of a cheat that you can use for the transitional epithelium is it's always going to have some kind of big hollow space in the middle. Now, some other tissues might have that also, but when you see this big kind of spiky hollow area and you're kind of stuck and you don't know for sure which tissue you're looking at, and you know you're going to have to guess, that's a good guess. It's transitional epithelium, because this area is what will need stretched as it fills with urine. Now we move away from the epithelial tissues and we're going to start looking at the connective tissues. We're going to look at connective tissues proper in this lab, and then in lab 5 we will look at the remaining connective tissues, which are the cartilages, bone, and blood. So here what we have is areolar connective tissue. Sometimes it's called loose connective tissue, but areolar is what you'll hear most often. And when we look at this slide, it's often really, really faint. It doesn't hold stain very well. This one is one of the darker stained areolar slides that I could find. And when we look, most of what we're seeing is extracellular matrix, and that's going to be true for all of the connective tissues that we look at, because that is something that makes connective tissue connective tissue. There are cells that are scattered, which is, we see the nuclei of the cells, and there's not too many of them. Most of what we see is extracellular matrix, and then we also see fibers. And that's going to be how we can determine which connective tissue is it we're looking at. So for areolar connective tissue, what we see regarding cells, we're actually seeing the nuclei of the cells. And those cells are called fibroblasts. Now remember, if you've already watched the chapter 4 lecture video, when you see blast at the end of a word, it means to make. So fibroblasts make the fibers that we're looking at. And when we're looking at areolar connective tissue, there's two types of fibers that we can easily see. These very, very thin fibers that almost look like cracks in the slide or like hair, those are called elastic fibers. And then in this example, they're fainter but thicker. We see running in the background, these are collagen fibers. So collagen fibers, fainter but thicker. And then the dark, thin fibers are elastic fibers. Both of them were made by fibroblasts. So like I said, this is areolar connective tissue. And areolar connective tissue is found just underneath the epithelial tissue surrounding practically every organ. It's just underneath the epidermis of your skin. It's just underneath the epithelial tissue that wraps the organs in your abdominopelvic cavity. It surrounds most of your organs just underneath the epithelia. And its job is to hold fluids, and it cushions the organs that it surrounds. If you've ever, bumped your arm and you get a knot on your arm, it is the areolar connective tissue that's kind of swelling with fluid and acting as a shock absorber right after you bump your arm. So it holds fluids, it moves fluids that are found in the tissues, but it's there as a protective barrier around your organs. Here we have reticular connective tissue. And reticular connective tissue is kind of like scaffolding. If you've ever watched a large building being built and they put up like the girders that support everything in the building, that's kind of like the job of reticular connective tissue. And we find it in places where there needs to be some sort of support, like in your bone marrow or in your lymph nodes or in your spleen. And it acts as a framework for the very soft inside of those locations. And here we find just a few, we can see some nuclei scattered here and there of the fibroblasts. And then they're kind of hard to pick out, but we see some fibers here, and these are reticular fibers. And those reticular fibers really are what are providing that framework. And I don't see any in this example, but it's common to see various types of white blood cells, especially macrophages and things like that in this tissue. But as a general rule, when you see the nucleus here and there, really what you're looking at are fibroblasts. And again, those are what make those reticular fibers. This tissue is adipose tissue, which is really what we generally just call fat, but really it's called adipose tissue. And it's another one that stains really, really faintly. But what we're looking at here, these large, roundish structures, those are the adipose cells called adipocytes. And these adipocytes, most of the cytoplasm and the nucleus are pushed off to one side, like we see right here, or like we see right here. And this large open area is a fat droplet, and that takes up the majority of that cell, that adipocyte, and it pushes everything else off to one side. So the job of these cells, the job of this tissue, is to store energy in the form of fat droplets. And additionally, by filling these cells up, they kind of act like pillows, so they can work to act as a cushion. And we'll often find them around certain parts of your body that need cushioning. So energy storage and acting as a cushion are the functions. We find adipose tissue in the abdomen, just underneath the skin, really just underneath the skin, in the whole body, but especially dense in the abdomen, in the buttocks, in the breasts. We also find adipose connective tissue around the kidneys and behind the eyes. So all of those areas you would find adipose connective tissue. The next two tissues kind of go hand in hand. They are both called dense connective tissues. This first one is called dense regular connective tissue. The next one we look at will be dense irregular connective tissue. And what we see when we look at them is lots and lots and lots of collagen. That's all of this pink stuff right here. And this is how you're going to know whether or not it's regular or irregular, because in both cases, the collagen is packed really tightly. That's the dense part. In dense regular, which we see right here, the collagen all runs the same direction. We can see all of these collagen fibers are running roughly parallel to each other. We'll see something different when we look at dense irregular in a moment. But one more thing that you see on this slide, these purple areas right here, these are the nuclei of fibroblasts. And remember, the fibroblasts are what make these collagen fibers. And the nuclei are kind of elongated because everything is packed so densely that they're kind of squashed. So the cells, the fibroblasts, are elongated. When we have the collagen running parallel in this way, we have to think of really what function would that serve. Collagen is a really strong fiber. It's a protein, and it's really, really strong. And if it runs all in the same direction like this, what we end up with is resistance to tension that goes all in the same direction. For example, when we have the tendons that join muscle to bone, then every time that muscle contracts, it pulls on that tendon, but that pull is always in the same direction. So we find dense, regular connective tissue in tendons. So here is our dense, irregular connective tissue. And again, we see collagen fibers, and it's packed pretty densely, but they are not all running parallel. Really what we're seeing is collagen fibers running all different directions, and we're seeing cross sections of most of them, but they are not organized nice and neat like we saw previously. So again, collagen really, really strong, and it's able to resist tension, but since the fibers are running in all different directions, then that means it can resist tension from all different directions. And we find this in the dermis of our skin. It also forms the capsules that surround most of our joints, any place where there's going to be tension in more than one direction. Here, our nuclei are not squashed. They're more rounded, like we would expect to see in most other tissues. And that's again because everything's not running the same direction, squeezing everything down. But these are the nuclei of fibroblasts. And the last tissue that we will look at for this lab is elastic connective tissue. And elastic connective tissue we find several places throughout the body, but the best example is in the walls of your blood vessels. And when we look at this, we see all these really dark lines, and those dark lines are elastic fibers. And we can see that those elastic fibers are kind of wavy, kind of folded, and that's because this tissue is unstretched. This is, again, the wall of the blood vessel, and if we were to stretch the wall of this blood vessel, then instead of having this wavy appearance, those elastic fibers would be straighter. Very similar to like an elastic waistband. If you stretch it, then it can kind of snap right back. And that's exactly what this tissue does in your blood vessels. It allows your blood vessels to expand, and then it brings that blood vessel back to its regular size. So that's the... Hello again, everyone. Today we're going to be looking at the slides for cartilage and blood. These are going to be the slides that are on Lab Exam 2. Now there's also the bone tissue slide, the osteon slide, which is actually covered in a previous video when I was doing bone structure. So if you haven't seen that, be sure to watch that video because it does contain a slide that could be on Lab Exam 2. So let's go ahead and dive in. This first slide is one that you've seen before back when we were first learning how to use the microscope. This is the blood smear, and what we're seeing here is what blood looks like under a microscope. And you'll know from lecture and just from things that you've heard before, the function of blood is to transport various things around the body, such as oxygen and carbon dioxide, and also nutrients and waste produced by the body tissues. So a couple things that we can see in this slide. First, the most abundant thing we see are these things that look kind of like little pink donuts or Cheerios. These are the red blood cells. Actually, we're going to call them now erythrocytes. Erythrocyte is the proper name for a red blood cell. Erythro is a root word that means red, and erythrocytes transport oxygen around our body. The other things that we will see are these cells that have these purple blobs in them, and they will all look pretty different from each other, but these are our white blood cells, and their proper name is leukocyte. Leukocytes are white blood cells, and these cells, you'll learn next semester in Bio 139, actually have several different functions because there are several different types of leukocytes, but for right now you don't need to know the types. But the thing that they all have in common is they are part of our immune system, so these help keep us healthy. And the final thing is all of this clear background area that's not one of those two cells. This is the extracellular matrix of blood. Remember, most connective tissues have a very extensive extracellular matrix. Blood is a connective tissue, so it also has a very extensive extracellular matrix. It's just in blood, we give it a more specific name. We call it plasma. So this clear background area here, this is blood plasma. All right, and now on this image, we actually see all three of the cartilages that you're responsible for knowing on this lab exam. And the reason that I have them all together is because the first thing that I want to do is point out how you can distinguish between the different cartilage types. Now, the thing that they all have in common is they all contain cartilage cells, and cartilage cells have the name chondrocytes. Chondra just means cartilage. Chondrocytes are cartilage cells. And just like we saw in bone, chondrocytes sit within a depression called a lacuna. So this is a chondrocyte in lacuna. Here is another chondrocyte in lacuna. Now, the way that we can tell the difference is we're going to look to see what do the fibers look like. And there's a lot of collagen fiber in cartilage. Over here on the left, this is hyaline cartilage. Here in the middle, this is fibrocartilage. And over on the right is elastic cartilage. So how do we tell the difference in those three? The fibers in hyaline cartilage are not easily visible. They're there, we just don't really see them, and under a microscope, hyaline cartilage, I've always thought, kind of looks a little bit like stained glass. Now again, just like we saw back with our first lab or the first of the tissue labs, I believe it was lab 3, don't learn colors, because colors can change from slide to slide. So even though it's pink in this image, it's often stained blue. I've seen it stained green. So don't learn colors. Learn to see what do the fibers do. And in hyaline cartilage, we don't see the fibers, and the cartilage looks kind of like stained glass. In fibrocartilage, the fibers all tend to run roughly parallel. Here we can see bundles of fibers all running parallel. And in elastic cartilage, we can see that the fibers run all different directions. So hyaline cartilage, you don't see the fibers. And then if you do see the fibers, if they run parallel to each other, it's fibro. If they run all directions, it's elastic. And as far as functions go, hyaline cartilage, its function is to give structural support, such as we find in the costal cartilage of the ribs. It also covers the ends of long bones at joints. For fibrocartilage, its function is it can resist compressive forces. So we find it in places like the discs in between your vertebrae, the intervertebral discs, where the two halves of the pelvis meet at the pubic symphysis, and also in those meniscus that we find in the knees. And over here on the right, elastic cartilage, its function is to provide a great deal of flexibility, and we don't find it very many places in the body. It's found in the throat in a little flap called the epiglottis. The epiglottis bends when we swallow to cover up our trachea so that food or liquid does not go down into our lungs. And then also in the stiff portion of the external ear, so the upper part of your ear where it's a little bit stiffer, not the lower part, the lobe, but the upper part of your ear that you can bend and it's very stiff and it snaps back. So that's where we find elastic cartilage.